welcome to Converging Dialogues. This is Xavier Bonilla. On this episode, I am very honored and pleased to have the very brilliant Siddhartha Mukherjee on the podcast. Siddhartha is an assistant professor of medicine at Columbia University. He's a cancer physician and researcher. He's a Rhodes Scholar. He has degrees from Stanford University, University of Oxford, and Harvard Medical School. He's published numerous scientific papers in scientific journals. He's also a writer at The New Yorker. He is the author of a handful of books, namely The Emperor of All Maladies, a biography of cancer, which won the Pulitzer Prize in General Nonfiction in 2011. He is also the author of The Gene and Intimate History, which was a New York Times bestseller. And he is the author of the new book, The Song of the Cell, an exploration of medicine and the new human. And that's what we talk about in this conversation. We begin the conversation by talking about what is a cell? How do we define a cell? We talk about the importance of Leeuwenhoek and Hooke. These were two early researchers and scientists. And they are essentially credited with not discovering the cell, but definitely observing the cell. We talk about the 5 basic principles of cell biology. We talk about the anatomy of a cell. He has a very nice visual aid of using a type of postal system to explain how the inside of a cell works. We talk about how important B cells and T cells are for the innate immune system and for the adaptive immune system, and how they're obviously useful for fighting off diseases such as HIV and COVID-19. And then we end the conversation a little bit by talking about stem cells and their continued promise. This is some work that he's done himself as well. It was such a pleasure to talk with Siddhartha, as I say in the conversation. Anyone that's read his previous books and read the new one will see that he is amazing at making, genes or cell biology super attainable, right? This doesn't read like a textbook, and he has such a way with giving good visual images and analogies to help us explain the things about us, what makes us human, and how we can better understand who we are so we can make more advancements in the future. So now I bring you Siddhartha Mukherjee. I am here with Siddhartha Mukherjee. Siddhartha, thanks so much for coming on the podcast. I'm greatly looking forward to this.
My pleasure. Thank you for having me.
Yeah, of course. You have written a fantastic new book called The Song of the Cell, an exploration of medicine and the new human. And so it's wonderful. Obviously, all of your books are wonderful. Many people will know The Emperor of All Maladies and The Gene. Is this a quasi sort of trilogy loosely, or no, they're just totally unrelated?
Well, it is a trilogy. I mean, they're related and unrelated. I think they're obviously bits of both. In a strange way, I think, the trilogy, if you think about it, begins with the gene, because that's the fundamental sort of unit of information. That information gets enlivened by the cell and the physiology of the cell. And then from the physiology of the cell and medicine, we turn to the physiology of the abnormal cell, which is the Emperor of All Maladies, which is also in some ways, of course, its own history of cell biology. So all the pieces are in there, but I think, it's a trilogy in reverse, as you want to think about it. But that's at least how I imagine it.
Yeah. So for folks that may not know you, just briefly give the 30-second biography, who you are, your background.
So I'm an oncologist, cancer biologist, and a cancer doctor, associate professor at Columbia University. My laboratory works on a broad span of things, including stem cells and immunology, particularly in blood cancers. I am the founder of multiple companies which are really involved in bringing novel medicines into existence. So, and what else can I tell you about myself? I think that's the high level biography.
Well, you're also a Pulitzer Prize winner and a New York Times bestseller and all of those small things as well. So,
So thank you for including those things.
Yeah, yes. So obviously you've done some really fantastic research and you've written some wonderful books. So let's talk about the new book. I guess you were kind of alluding to it. So why did you want to study the cell? Just kind of go all the way to the very the origins here. What is a cell? How do we define it? And then why did you want to write about this?
Well, the cell is the single smallest autonomous unit of life. I felt that we think about our bodies, and for the longest time into the 15th and 16th centuries, we thought of our bodies as continuous. What I call flesh. No one knew what flesh was made of. It's only when scientists like Robert Hooke and Leeuwenhoek looked down the microscope, they began to realize that in fact we were made out of these single autonomous units, separate autonomous units. And what's amazing about them is that they are living units. They are the least, they're the smallest living units that we can find, ranging from bacterial cells, which are also cells, all the way to massive and complex neurons in our brain. These are all autonomous units. They function independently. They can often assume very diverse functions. Again, imagine the difference between a white blood cell that guards you against infections and a neuron that fires, when you have a particular sensation. Both of those are cells. The reason to think about cells and to really start thinking about cells is that, the last, I would say, almost century has been preoccupied by genes and genetics. And of course, genes and genetics, the iconic image of DNA is absolutely essential to understanding life, understanding ourselves, understanding medicine. But I think interestingly, the genes are lifeless without cells. And so the whole idea here is that the lifelessness of genes is enlivened by a cell, and only at that level can you understand life. You can read the Morse code of DNA, but what you won't get from that Morse code is very much about sort of what life really feels like or looks like or behaves like. And that's really important, because if you want to understand medicine, you not only want to understand that source of information, that's crucial, but you also want to understand how that information is enlivened. And I often tell people, by reading the code of genes, it's impossible to figure out what I call emergent properties in this book, the properties that cells acquire when they integrate all this information. A cell is like the massive integrator of all this information. Without that integration, there would be no life. And what's even more astonishing is that each cell integrates information differently. And maybe an analogy helps here. You can imagine our genetic code, our genomes, as a symphony written on a piece of paper. That symphony is soundless. It is lifeless. It's only when an orchestra plays that symphony when it becomes a song, hence the song of the cell. But also the information. What's interesting is that each cell type in your body is playing different parts of the symphony, even though the score handed to it is basically the same or identical, or nearly identical, I should say. But each cell in your body or cell type in your body is handling that symphony differently. And when it goes off key, when the tunes don't sound right, then the dysfunction that results from them is a cellular dysfunction. It goes back to the central theme of the book, which is a very brave statement made in the 19th century by Rudolf Virchow, who's a central figure in the book, who makes the argument that all diseases, and I'm going to repeat that, all diseases are cellular diseases. To hunt for a cure or treatment or to hunt for the cause of a disease, you can certainly look at genes and genetics, but ultimately it's the location of dysfunction, the locus of dysfunction is going to be the cell. It's going to be a cell that's somehow not working correctly or invading another cell or causing an infection or whatever it might be. It is the cell, the living unit that goes wrong in all diseases. And so that brings me to the book, which is, in order to understand ourselves, in order to understand normalcy and pathology, we need to understand cells. And that's what the ambition of this book is to remind ourselves that we're moving from overlapping centuries of the gene to the overlapping century of the cell, and we're finally trying to sort of step up the rung of complexity in our understanding of life towards understanding the song of the cell biology.
Yeah, you describe it, masterfully. I mean, your analogy of the symphony is a good one. And just as an aside here, one of the things that many people, I think, will really get out of the book is that, writing about a cell is, it's not like it's a cellular biology textbook. You have so many wonderful ways of describing, just as you did there, why this is important and why this is really essential to understand, yes, disease, but then other things about life and about how we're navigating through the world and moving through the future, which is wonderful. You talk about, you mentioned them, is it Leeuwenhoek and Hooke for observing the cell? And this was back in the 17th century. Can we say that they kind of discovered or observed, a cell as its own entity, or just maybe talk about the significance of them observing it?
Yeah, I mean, they're fascinating characters who open the book, and there's so many. But Leeuwenhoek and Hooke are in some ways opposite characters, and they, within a decade of each other, discover cells. They're total polar opposites. So let me tell you a little bit about both of them because I'm so interested in their lives. I should tell you.
Yeah.
I used to live in the when I was at Oxford. Funnily enough, this was years ago when I was still a graduate student studying immunology. I lived in the building across where Robert Hooke lived.
Oh wow!
There was a little. I used to pass by Hooke's house, which is of course kind of a national monument of sorts. Mm-hmm. But let's talk about Hooke first. Hooke was a child of indigent parents, came on scholarship to Oxford, worked as an assistant to another scientist. But I describe his intelligence as phosphorescent and elastic, like a rubber band that glows when it stretches, because he could, and there's a pun in there, of course, because Hooke's one of his greatest contribution is his understanding of elasticity. And so he was the rubber band that stretches. In fact, we still use his equations to understand how elastic materials are elastic and what they are. But anyway, he was just a foraging intellectual, a wild intellectual in the sciences. No science was outside his reach. From physics to chemistry to biology to even architecture, kinetics, motion, planetary motion, astronomy, telescopes, microscopes. He did everything. And he was, of course, for a while, the president of the Royal Society. So he is this character, and he looks down a microscope, in his case, a compound microscope, and he looks at a slice of tissue and finds, to his utmost surprise, that it's not continuous. It's made out of what he calls a great many little boxes. And he's looking at plants, so it's mostly cell walls that he's looking at or finding. And he draws them, and he publishes this book, which becomes a kind of big hit of the year, in which he describes these great many little boxes. And he's thinking about a name for them, and finally he says, well, they look like monk's chambers, so cella or cell. But he doesn't fully appreciate the extent of his discovery or how he's changed, sort of how much he's changed biology. I will come to that in a second. Leeuwenhoek, who's comes a little bit later, is quite the opposite. Leeuwenhoek's not a scientist, not trained in science, doesn't have the vocabulary of science. He's a cloth trader, a cloth merchant. He's in the Netherlands. And he wants to look at the quality of thread. And he has a kind of obsessive, secretive, reclusive quality about him. But he wants to examine every thread that he trades, making sure that it's that the silk is real silk and not mixed in with other things and so forth. So he invents his own microscope. His microscope is a single lens microscope or a simple microscope, but they're beautiful. Even he makes about 500 of them. They're mounted on brass plates with tiny screws to move the object so that you can get it into perfect focus. And very soon he stops looking at threads and says, what about rainwater? What about he picks his teeth, and what about the things that live between teeth? He looks at his, sounds very bizarre. He looks at his own semen. And in every case, he finds what he calls animal cues, these independent seeming organisms that have life. They're moving around. They're doing things. And so he starts drawing them. He starts writing letters to the Royal Society, which actually Hooke himself receives. And very soon they have a correspondence with each other. But Leeuwenhoek is quite the opposite of Hooke. He's secretive, he's reclusive, his language is colloquial. He doesn't even know the extent of what he's discovered, but he's obsessed by it. So together they discover these things, animal cues or cells. And it's only much later, almost a century later, that scientists begin to realize that these things are the universal common unit of all living beings, and that's the foundation of cell theory. And that's when Schleiden and Schwann, two scientists looking at animals and plants, a zoologist and a botanist, realized that this is not something bizarre that they discovered, that Leeuwenhoek and Hooke discovered in rainwater and other entities. This is a universal principle of all living beings. They're all made out of cells. Animals, plants, everything is made out of cells.
Yeah, it's so powerful, almost knowing, like the fact that, you discover something or you observe something and not knowing the full extent of how it is the essential building blocks for life is pretty powerful. You talk about in the book 5 principles of cell biology, which are living organisms, or all living organisms are composed of one or more cells, that cells are the basic unit of a structure. All cells come from other cells. Normal physiology is cellular physiology, and that, as you mentioned earlier, disease equals disrupted physiology of the cell. Maybe just kind of talk through some of those 5 principles, how we came to them, and why they're so important.
Yeah, so they're the basis for virtually all cell biology. Cell biology you can think about as a very broad field, and remind me to talk about the challenges of writing this book. Just in terms of the writerly challenges, but so the first principle, that's Schleiden and Schwann figuring out that as I said, the book begins with this moment which is recorded in a very slight way, in just one letter. Schleiden, many years later, it's a very tiny sort of reference, but I loved it. And found it so evocative. So they're sitting down in Berlin one evening, and I think Schleiden or Schwann turns to Schleiden and says, I've been watching animals develop, tadpoles and other animals develop, and I've been looking at it, and their development, there's this thing which later becomes recognized as a nucleus. But there's something in that development which suggests to me that there's a kind of organization to that development. That these units, he doesn't know where they're coming from, that these units, they coalesce, as he calls it. Again, he doesn't understand the whole process, but he does understand that the animal is being built out of units. He doesn't know where the units are coming from. He knows their cells, but he doesn't know where they're coming from. He doesn't have any claims about what their function is or what they do. But he has a vague sense that this animal is an assemblage of the very thing that Hooke had described, a great many little boxes that Hooke had described. He calls them elementary particles. And the botanist says, well, wait a second, I've been looking at plants, developing plants, and I also noticed that there's a similar sort of organizational system. And there's sort of, this is all been happening in Berlin, dusk is falling, and they all of a sudden realize that they might have discovered some universal principle. And so the next day, they go to their microscope and then immediately look down the microscope and find that in both animals and plants, that there's a common means of formation, as they call it, which is through cells. So they lay down the first couple of tenets of cell theory. Number one, all organisms. It's a very audacious thing to say.
Oh yeah.
All organisms are built out of cells, and that's the first principle. Then we move on to the second principle. Which order did you go in?
I think I use the same as in the book. Cells are the basic unit of a structure.
That's right. So these are this is again Schwann and Schleiden saying that it's the basic unit of how an organism is built. So it follows out from what they describe. But what's interesting about Schwann and Schleiden is that despite this incredible insight that they have into the fact that all organisms are built out of cells and there's that's how they form their structure, they can't get themselves to figure out how cells appear.
Mm-hmm.
They are they're in the lab of a very influential figure whom we've largely forgotten, Johannes Müller, who's also in Germany. And Müller is sort of caught. I would say he's a kind of a vitalist soft, and by that I mean that he's caught in two worlds. There's a world around him in biology that believes that there are vital forces that are unique to life, and so the appearance of a cell, if ipso facto, would be a consequence of one of these somehow this vital force. This is in contrast to others who believe that there are no such things as something special or biological vital force. It's all physics and chemistry brought together, as we'll find out later. Mm-hmm. That's actually true. So Schleiden and Schwann keep thinking that where do cells come from? Then they just can't grasp it. And in fact, they turn to the theory that by analogy, they say crystals crystallize out of solution. So cells must be crystallizing out of this vital fluid. But especially Schleiden knows that there's something wrong. It just doesn't make sense. So the world sits there until several people, among them most prominently Virchow, but others actually preceding Virchow, make just the opposite argument. They say, well, it's so simple. Cells come from other cells. There's one cell, it divides, makes another cell, it divides, makes more cells, etc., etc. So that's the Virchow, and in fact, he's preceded by a couple of other scientists. But Virchow really puts it on the map by saying omnis cellula is cellular. Only from cells come cells. That's again a universal principle of cell biology. Every cell in your body, whether it's going to make new cells or not, is irrelevant, but has arisen from the first cell in your body. That is an absolute truth. The first cell in your body, every cell that you have currently in your body can be traced back in its lineage to the first cell in your body. If we could light up that cell with a molecular beacon, your whole body would glow because every cell would be derived from that cell.
It's a fascinating prospect of thinking of like this, almost like this infinity circle of like just cells reproducing and making, and they all come from other cells. They don't, there's not one that's kind of patient zero, if you will, I guess maybe back in time.
Patient zero is your fertilized egg.
Right.
So that's Virchow. And then Virchow he doubles down and he says, more than that, normal physiology is the consequence of cellular physiology. Again, an incredibly audacious statement. You could think that in the late 1800s, you could imagine someone saying normal physiology is because of miasmas or a psychic, or as the Galenists believed, it's how four fluids, phlegm, red blood, black bile, and yellow bile are balanced in your body. That would be the Galenic belief. And here is Virchow saying, no, that's all wrong. Everything that your body is doing as an organism is the consequence of what cells can and cannot do, the function of cells. He's reflecting back on again a really hidden character in medical history, a Frenchman named François Raspail, who of course is given his due in this book, but Virchow is picking that up, and then finally he makes the most audacious statement of all, and he says, if that's true, if normal physiology is really a consequence of cellular physiology, then its opposite must also be true, which is that whenever you think about disease, what you should really be thinking about is abnormal cellular physiology.
It's just so interesting how there's like these, one person makes a observation or a discovery, and then the next person is picking it up, and how there's these universal claims that are made and how they still hold, which is really powerful. And obviously you weave the wonderful narratives in play throughout the book. I want to talk to you about some of the other aspects later in the book that you talk about. So before we do, just kind of give a reminder of sort of the anatomy of a cell. In the book, you use a really nice postal system analogy to explain the kind of organized cell. So maybe just kind of use that with talking about proteins and ribosomes and RNA and all that.
Yeah. So before that, let me talk a little about the challenge of the book. And
you mentioned that. Yeah,
how this book is written, because I think your listeners might find that interesting. When you write a book of this length, it's almost 500 pages. The general rule is chronology is your friend. It is your best friend. So you follow time, and especially in science and writing about science history, you follow time. And time helps you organize the book, because this happened in 1835, that happened in 1846, because people are building discoveries on top of discoveries. The problem with cell biology, unlike, for instance, genetics, or unlike the history of cancer, so the problem with writing a book in a chronological way about cell biology is that the discoveries sort of happen almost in parallel, and there's so many different cell types, is that people are asking very different questions. Some people are asking how blood works. Some people are asking how the immune system works. And so it would be a very, I tried it. It would be a very tiresome book. You would have to shuttle around just as cells do, because this is not the story of, this is not a mystery story like Emperor of All Maladies. It's not a plot thriller that ends with how do we find treatment, prevention, and cures for cancer. This is a story about what is going on and how do we imagine the body. How do we learn about the body, and how do we treat cellular diseases? So the challenge in writing the book was how to do that. So what happens? The book has chronology up to about Virchow and a little bit beyond that, but then after that it changes, and each individual chapter becomes a cell. It's a little bit hard to explain, but it takes on a cell as if it's an individual chapter, and uses that chapter to highlight a particular feature of life that cell highlights. For instance, communication, autonomy, homeostasis, etc. But we begin that journey to some extent. We begin that journey by first asking how it is that we sort of begin that journey by sort of taking a journey or a trip into a cell. We sort of enter we enter a cell as if it was docking a spaceship. So we go into this kind of what I call the spaceship cell. You board the cell, and then you go inside. And the first thing you discover is that there's a whole anatomy inside, just like you can imagine an anatomy of the human body. That a cell is full of anatomy.
Right.
And as I point out, that anatomy is what gives the cell autonomy. So let's start from the outside. There's a membrane. Every cell has a membrane. It's bound by a membrane. The membrane is very special because it has pores in it that allow things to go between one cell to the next cell, signals of all sorts, nutrients, passages in that cell membrane. And then you dive into it, and then all of a sudden there's this highly organized system or highly organized fluid called the cytoplasm. Mm-hmm. One of the main things that a cell does is make proteins. And the reason, or at least live cells do, and the reason that they make proteins is that these proteins are the workhorses of the cell. They are the ones that are going to do all the hard work. They're the enzymes that make life's reactions possible. They are the center of signals that pass between cells. So you have this whole system. So an enormous question in the 1950s was, how does all this happen? Right. And the work of people like George Palat, I won't mention all the names, figured out that the cell actually has an incredible apparatus. Virtually every cell has it. To make proteins and deliver them to the right place, because they have to go to the right place to perform their function. So I describe it like a postal system. So first, the message to make a protein comes from the nucleus of the cell. That message, of course, is stored in DNA. It's the genome, and the genome is in the nucleus of cells that have nuclei. Then it's made into what I call a soft copy, the letter. Mm-hmm. That's RNA, a long strand. Like DNA, it's a copy, a soft copy. That copy gets written into a protein, just like a letter writer. And the letter writer of the cell is the ribosome. The ribosome is a massive complex of proteins and RNA, which is like a letter writing machine. It'll clamp down on the message, the RNA, and write the letter, which is the protein. So now the protein can go in many places. It can go someplace in the liquid of the cell, or it might be bound to go to the membrane to form one of those pores we talked about, mm-hmm. or it might go into someplace else. So the first place it goes, if it needs to be exported, is usually at this structure, which I actually draw in the book, called the endoplasmic reticulum. Sounds like a long name, but really is like a lacy series of membranes. Mm-hmm. And the freshly made protein is delivered into the endoplasmic reticulum. And then from there, it matures. In the end, lots of things happen to it in that lacy membranous structure. It gets folded correctly. Sugars are added to it. It gets clipped. It gets matured almost. And I really talk about it like as if now the letter is being really packaged in its proper packaging form. It then goes to what's called the Golgi apparatus, another membranous structure. But so again, continuing the analogy of the letter now is being routed. That's the posting station. And then from the posting station, it goes either out towards, to make a signal to another cell. It could go to the membrane, or it could go to some other compartment inside the cell where it does its function. So that's the sort of letter analogy. But the bigger analogy, I think, which was really interesting to write and actually kind of fun to read, is thinking about the cell as a spaceship. Mm-hmm. Boarding the cell, going inside and looking around yourself and seeing all of these things happen, this internal anatomy of the cell. It's something that I love to think about as a child. What it would be like for me to become a tiny virus, say, or something very tiny that could go inside the cell, and that comes alive now in the book.
Yeah. You have a really nice way of giving a lot of these visual pictures and images to kind of make something come alive, something like, cell biology, which can be kind of, for some people maybe boring or whatever, but it's so vibrant when you're writing about it, giving these kind of analogies like a postal system or a spaceship. And obviously in the picture you just drew with the postal system, the proteins are obviously, like you said, the main workhorse. They're what's really essential. And so later in the book, obviously you talk about many things, which are, I really liked your bit on IVF and the miracle of IVF. You talk about gene editing, but I want to ask you, in this context, because obviously you talk about it in Emperor of All Maladies, but here you mention basically three major things. You talk about the role blood has in giving this kind of connective relationship. So obviously you talk about white blood cells, red blood cells, platelets, but you also obviously talk about T cells and B cells, which I think are obviously super, super important. And I think they have a lot of relevance, in general, obviously, but in terms of a pandemic we've been through with a virus, obviously they're hugely important for cancer and they're important for HIV and many other things. So maybe just kind of walk us through what B cells and T cells are, CD4, CD8 proteins in T cells, how all of these things are super important to understand for many of the diseases that people will have or what have you.
Well, so if you think about the immune system, it's broadly divided up into two parts. And we need to, they're two very distinct parts. There's some overlap between them. The immune system has one part, which we now call innate immunity. Mm-hmm. And that is a kind of immunity that's sort of the first responder. Monocytes, neutrophils, macrophages, some cells you may not have heard of, but they're the first responder cells. They are the ones that rush in to fight an infection, and they're very, very, very important. But what's different about these innate immunity cells is that they don't have a learned capacity. They don't learn anything about an infection. They just go fight it. And by learn, I mean they don't have any memory. The next time the infection comes, it's not as if that neutrophil is going to remember that this was the virus that it encountered the first time around. The other wing of immunity, which is quite different, and actually appears later in evolutionary history, is called adaptive immunity. And as the word might suggest, it has the capacity to learn and remember. It's adaptive. It adapts itself so that the next time you encounter that same virus or the same pathogen, it's actually learned something about it and remembers it. And that is what protects you from repeated infections by the same pathogen. It's adaptive immunity. Now, adaptive immunity comes in two halves, and that's what you were talking about, B cells and T cells.
Right.
B cells are cells that are, as I said, part of adaptive immunity, and they make antibodies. Antibodies you can imagine as missiles or harpoons that are made by B cells, and they go and harpoon and kill pathogens as they come. They can neutralize them. They can prevent them from going and infecting other cells, and they're absolutely important in anything. And that's why during the COVID infection, everyone was talking about what is how many antibodies you have, right. What antibodies you have. That's because these are the proteins made specially by B cells that when a pathogen comes, these antibodies will go and either attack and kill it or neutralize its capacity to bind. A virus's capacity to bind to a cell and enter it. So that's a B cell. And what's amazing about it is that it has memory. So as the B cells mature, once they've encountered the pathogen and they've been appropriately stimulated, as they mature, they convert themselves into professional antibody secreting cells called plasma cells, and also into a compartment of cells or kind of cell called a memory B cell, which keeps the memory. It has that same antibody and it stands by on the ready so that the next time you get an infection, you'll already have a memory. That's why, again, adaptive immunity, you already have a memory of that infection. And that's why antibodies are so important. If you don't have the antibody, there's no memory B cell, or if a virus mutates and the antibody, the harpoon can no longer harpoon that virus because it's changed its own structure through a mutation, then of course you could get reinfected by a virus. And that's what's been happening, as we've seen in the COVID pandemic. You can raise antibodies against one kind of virus, the virus mutates, and although you have memory of that prior infection, the antibodies don't work against the new infection. And you can quickly figure out why this is important for vaccines. So in making a vaccine, you're essentially priming a B cell before the pathogen even arrives, telling it to make the antibody, and that antibody is sort of lying in wait, and the B cell is lying in wait so that when the infection actually hits you, you're already prepared. You're an army that's already prepared. You've already seen the face of the enemy before you get it. Which brings us to T cells. T cells are again a part of adaptive immunity, but they don't make antibodies. They don't make the missiles. They don't make any of that. What T cells do is that they're surveillance drones. They go around the body looking at cells. They come in two basic kinds. I'll talk about that in a second. But the ones that are the killer ones are drones, are surveillance drones. They don't make missiles. They go around the body sniffing each cell and asking the question, is there a virus hiding inside you? And they do it through an elaborate process that I describe in the book, but that's basically what they do. They are the basis by which the body recognizes what is called non-self, things that shouldn't be there inside a cell. And what's interesting and important is that they're looking for things hiding inside a cell. A B cell is looking for a pathogen floating around in the blood and making an antibody to that. A T cell is looking for something that's hiding inside a cell. That's why I call them surveillance drones. It's got a unique capacity to go and sniff around and do that kind of surveillance. T cells come in many forms, but two very important forms. One is what I call what is called the traditional killer T cell, and that brandishes a protein called CD8 on its surface. And then there's another kind of cell which is called a helper T cell. Although, as I say in the book, calling it a helper T cell vastly underestimates its function. It's like calling Thomas Cromwell a bureaucrat. It's a helper T cell is sort of a mastermind, a master functionary. It doesn't generally kill cells, but it also has this capacity of detecting an infection. And when it detects an infection, it's the orchestra coordinator. It is the hub through which the B cell and the killer T cell and the innate immunity are all sort of coordinated through chemical signals. And without helper T cells' help, a B cell would never be able to mature, your vaccines couldn't work, etc. Why is that important? Because they carry another marker on their surface called CD4. And it's, I'm sure most of your audience knows by now, the HIV is a disease in which these helper T cells are infected. The CD4, you remember people talk about CD4 counts. That's because HIV infects helper T cells, and the entire T cell help system collapses. And when the T cell help system collapses, your whole immune orchestration collapses, and you can no longer fight infections the way you normally fight infections.
So I was going to say, so this is kind of as a standard how B cells, T cells work, among other things. Of course, we understand that each individual organism, each individual person has a different biology, different ways in how these things do fight against certain viruses or does not fight well against certain diseases or viruses. So in this way, I mean, this is a very kind of general question, but if we understand these things about aspects of adaptive immunity and kind of the initial innate immunity, what makes it, I guess, difficult for certain types of diseases or viruses such as SARS-CoV-2, such as HIV and cancer? Obviously, these are unique in their own ways, but is it just a breakdown? Is it just that there's not enough that's produced, or we just don't know enough about the virus or disease and we have to learn more? How do these things, I guess, not do their job well or not enough for some of these diseases or viruses generally?
Well, we're finding out more and more about this as we move along. This goes back to Virchow. The pathobiology of a virus, the pathogenesis of a virus, really depends on its ability to disrupt cellular function. Even infectious diseases are really ultimately diseases of cellular dysfunction. Different viruses do it different ways, and they're all very devious about it. The body is devious because it can, of course, raise antibodies and T cells against a virus, but different viruses do it this way, different ways, and we're finding out how. I'll give you a couple of examples. I just talked about HIV. HIV cripples the immune system by infecting the very cells that coordinate the immune system. So those are the CD4 cells. Very different from HIV is a virus like SARS-CoV-2. Now, SARS-CoV-2 we're still understanding, and there's still things about it that are still emerging. But
Right.
one way that SARS-CoV-2 seems to be so pathogenic to some people is that it disrupts the capacity of a cell, of an infected cell, to send out the first signals that it sends out to the innate immune system. Remember, I said there were two kinds of immune systems. Most likely, the pathogenesis of SARS-CoV-2, and I'm saying most likely because we're still in the middle of figuring this out, but SARS-CoV-2 is very devious, and it manages somehow to become particularly pathogenic in either in individuals where this innate immune system is already a little bit weakened. It's not visibly weakened. They are not, they're walking around normally. We don't think that they're visibly weakened. But that's one thing, it exploits that system. And in other individuals, it actively, and we still don't know exactly how, but it actively suppresses what the initial alarm bells that a cell sends out as part of its innate immune system, innate response to the virus. Because of that, it doesn't allow a proper immune response to happen. As a result of that, we think that what happens is that the initial alarm bell that a cell is infected isn't sent out properly. But then in many individuals, there's a second alarm system, and that gets activated. And that's when an incredibly strong inflammatory response pours into the lungs. And it essentially, there's the fog of war. And it's really a sort of like your body fighting against itself. And that's responsible for all the fluids that pour into the lung, cause this kind of interstitial, almost like a pneumonia, and all the other things that happen as a consequence of SARS-CoV-2. So two completely different ways. The virologist Akiko Iwasaki has called it, for SARS-CoV-2, has called it immunological misfiring. It's as if the immune system fires, but it fires too late and then overfires, causing the real pathogenesis of the nastiest pathogenesis of SARS-CoV-2. Now, SARS-CoV-2 does a lot of other things as well, but I'm just talking about the immune system and COVID. Well, to summarize, different viruses use different mechanisms to infect and to potentially paralyze the immune response against them. Some of them we can effectively remove from our bodies, SARS-CoV-2 being one of them, influenza being another one. Some of them actually we can't even remove from our bodies, like Epstein-Barr virus causes, or herpes simplex virus, herpes. They've created capacities to hide away from the immune system using, again, very devious and different mechanisms.
So I want to ask just one last thing here real quick, because I want to respect your time. At the back half of the book, you talk about stem cells. Obviously, this is a massive topic, and you'll have to give me the condensed version here. But you talk about embryonic stem cells, human embryonic stem cells. Obviously, there's been some political things about this in the United States. There's certain ethics around this. But just talk about stem cells in general and the embryonic stem cells, and then you can kind of land us home here of what is the future understanding of the cell and various cells, and how that could be really important for our future evolution and development.
So embryonic stem cells are very, very unique because they have the capacity to do two things. One is that they have the capacity, as stem cells, they have the capacity to give rise to virtually all the tissue in an adult body. They can give rise to cartilage and bone and neurons and blood cells and so forth. But they also have a capacity to renew themselves, to give rise to themselves. And as far as we know, pretty indefinitely. So you can take a culture of embryonic stem cells on a petri dish, culture them, and they will keep giving rise to more embryonic stem cells. But if you change the conditions, they can give rise to all these adult tissues. So they are very crucial to understanding any disease, because you can derive any tissue, essentially, for the most part. You can derive any tissue from those cells, and you can also they're also crucial to understanding development, because as those tissues are being derived, you can ask the question, well, what's making it a cartilage cell? What's making it a neuron? You can essentially study the entire process. There's a special kind of cell which is more recently being described, called the induced pluripotent stem cell, IPS cell. This is a cell that using genetic manipulation, we can make it almost like an embryonic stem cell. A cell that's pluripotent, which means that it can give rise to multiple forms of tissue, bone, cartilage, what have you, neurons. And again, these are crucially important. They also give normally, you can, under certain conditions, they keep giving rise to copies of themselves. So it's a stem cell. A stem cell is defined as something that has the capacity to give rise to, or divide and give rise to, a copy of itself under certain circumstances, as well as a copy or differentiate into other mature cell types, as I gave you examples of, blood and neurons and others. And these IPS cells and ES cells are particularly important because they're not restricted to just being able to make blood components or blood cells or just neurons. They can do all. They can do virtually all cells. I shouldn't say all. They can make virtually all cells in the adult human body. So they're absolutely crucial to studying disease, studying how the body repairs itself, studying development, and studying how stem cells work in general.
Yeah, no, there's a lot of potential there, and so obviously we need folks like yourself to keep looking at many of these aspects. Siddhartha, this has been such a wonderful conversation. The book is called "The Song of the Cell: An Exploration of Medicine and the New Human." It's absolutely fantastic. I hope everybody picks it up. Any places where people can find you or reach out to you, or any of the best places?
Well, I'm doing a bunch of readings. Boston tomorrow. Not tomorrow, sorry. Boston, October 25th. New York, October 26th. Then I'm in the West Coast in early in November, and then in Miami, I think in December. So a whole bunch of places, whole bunch of bookstores. But of course, I'm on, always around. If people have particular questions, I love talking to booksellers in particular. So anytime any question arises, I have a website. It's easy to find me. I have a big Twitter account. So if there's a question, I always try to get to it.
Yeah, no, that's wonderful. Well, thanks so much for the conversation. It was a real delight.
Thank you so much, Xavier.
Thank you so much.